KJ, are you going to tell us when we're live?
You are live.
Oh, great. Okay, people are starting to trickle in. Excellent. All right, guys, it's 2 p.m. Welcome. We're going to go ahead and give it a second so that folks can keep trickling in. Excellent. Well, thank you so much for joining us this month for this month's version of What's Up in the Watershed. I'm really excited that we have Kirk Fusco here with us today from the Florida Department of Environmental Protection talking about horseshoe crabs. So just a reminder that this webinar is being recorded. So if you want to share it with friends and family after the fact, it will be available on YouTube. You will notice that the chat box feature has been disabled, but we definitely encourage questions. So if you have any questions for our speaker today, please go ahead and ask them in the Q&A section, and we will make sure that they get answered live or after the presentation. So without further ado, Kirk, we're just going to jump right into it. I'm so stoked to have you here because we're talking about horseshoe crabs, which is like one of my favorite animals in the lagoon. And I know that you have been doing a ton of work down on them in the central lagoon region, including some tagging of the acoustic nature, which I'm excited to hear about. So we will go ahead and get started. Kirk, thank you so much for joining us.
Thank you so much, Jesse, and thanks to the IRL NEP for having me. I heard that usually these talks are right around, I talk for 30 to 45 minutes, and then we have time for questions. Anyone who has ever had the misfortune of hearing me talk about horseshoe crabs knows that I am rather long-winded, but I try to keep it interesting for you all. Horseshoe crabs are a fascinating animal that I get super energetic and jazzed talking about, and hopefully with this presentation, you all will be a little more excited about them too by the end of it. So like Jesse said, my name is Kirk Fusco. I work for DEP, Department of Environmental Protection, state agency. Under DEP, I'm in the Office of Resilience and Coastal Protection. And under that little branch there, I'm actually part of the Aquatic Preserve Program. State of Florida has 45 aquatic preserves spread throughout the state. For those people who aren't as familiar with aquatic preserves, I always kind of say I think of it kind of like a state park, but underwater. So State Park Service is also under DEP as well. In terms of the Aquatic Preserve and Office of Resilience and Coastal Protection side, we do a lot of research and outreach, such as this, some restoration as well. So primarily, as you'd expect, for our division, it's all aquatic based. Most of it is saltwater, but we do have some freshwater aquatic preserves and programs as well. So jumping forward from there, of course, this is what you all hopefully came to hear about, which is the American horseshoe crab. So before I start talking about horseshoe crabs and how amazing they are, I like to give you all a little background and biology kind of to set the stage for us, right? So I find that I get most excited about learning about animals when I know interesting facts about them or just to have a basic knowledge of what they are. And unlike dolphins and sea turtles and manatees, what we call the charismatic megafauna in the scientific community, not as many of us are very well associated with horseshoe crabs. Horseshoe crabs, they are invertebrates, right? Meaning animals that lack a backbone. There are, the more I do outreach with horseshoe crabs, there's a lot of things about them that are amazing, but also very unknown. And then we also have misconceptions and things like that. They're in no way venomous. I've heard that many times. I'll go into their anatomy as well. But for our first jumping off point, let's talk about who are horseshoe crabs. So I always like to make the joke that, today we're going to talk about crabs. They're not crabs. Horseshoe crabs are not true crustaceans. So crustaceans would house the group with crabs, shrimp, lobster, a lot of other really cool marine invertebrates as well. But interestingly enough, despite our naming of them, horseshoe crabs are not crabs at all. They're actually more closely related to arachnids. Here on my slide, you'll see on the right-hand side that's called a phylogenetic tree. And I am a scientist who loves talking about what's related to what. So phylogenetics, that's just our fancy way of saying, hey, it's a family tree for different organisms, different species. So as you can see here, we would say that horseshoe crabs, as well as arachnids, as well as those crustaceans that I referenced, they're all under this one branch called arthropoda. And that just arthropoda, it just references animals with jointed appendages, so usually hard shells. Now, where do they differ from the crustaceans? Well, as we go down this phylogenetic tree here, you'll see that eventually, as we lead down, there's a split called Chelicerata. Chelicerata houses the chelicerate animals. What in the world is a chelicerate? Well, a chelicerate is actually an animal that has tiny little feeder arms. So they will actually, and I'll show you in some of the horseshoe crab anatomy slides, horseshoe crabs have these tiny little appendages that make them very unique from crustaceans. And then continuing, as I said, horseshoe crabs are much more related, closer related to spiders, but also ticks and mites. They all fall into that Chelicerata subgroup there. So continuing along with our horseshoe crab. So the species that we're talking about today, it's easy because there's only one of them that we will be discussing. So horseshoe crabs, they for our species, genus and species is Limulus polyphemus. So remember, we have that what we call binomial nomenclature. That just means, hey, as scientists, we like to name things with first and last name. So for the American horseshoe crab, makes my life easy because I only have to remember one species, unlike so many other animals. Birds, mammals, sea turtles, all of those have different species. So we only have, in North America here, we only have one extant species of horseshoe crab. That word extant, the way that I like to define that is just, it's essentially the opposite of extinct. So we have one horseshoe crab species in the US, in North America, that is currently living and existing, so not extinct. So continuing along in terms of that scientific naming, Limulus polyphemus, Limulus just means Latin for odd or askew. I feel like that's pretty self-explanatory with this animal, with the horseshoe crab. And then polyphemus also relates to unique eye structure, which I'll get into as well eventually. In terms of talking about, okay, cool, they're an extant species, right? We currently have them in North America. Well, where in North America are they? So they're actually found all along the eastern seaboard of the United States, the whole Atlantic, and then as we come south down to us in Florida, down the coastline, they're also found in the Keys as well, and they actually wrap around Florida, and we have them on the Gulf Coast of Florida as well. Horseshoe crabs are also found in Alabama, Mississippi, but there's a really interesting geographic characteristic where horseshoe crabs, they are found, panhandle area of the Gulf, so Florida panhandle, right? Those states that are closest to us, Alabama, Mississippi. But then all of a sudden, they're not found in the rest of the Gulf. Primarily, they're not found in Texas whatsoever on their coastline. That sounds, that's not too exciting, but the exciting part comes with the additional area that they're found. Horseshoe crabs are also found on the Yucatan Peninsula as well. So, in terms of horseshoe crab geography, I always find that fascinating that, okay, they're on the whole Atlantic eastern seaboard, right? We have them in a good portion on the east side of the Gulf, but then on the west side, nothing. And then they pick up again on the Yucatan Peninsula shorelines. In terms of a few other more basic foundational facts about them, they are benthic feeders. What that means is they're bottom feeders. They like to eat stuff that lives on and in the seafloor. So those things would include a lot of bivalves, right? So bivalve two-shelled animals. They love snacking on oysters and also clams, mussels, and then in addition, more what we would call infauna. So things like worms that live in the substrate. Some additional a lot of our clam species, they actually are really good at opening up bivalves. One of their appendages actually has a special spine that folks believe that that's pretty much like the main use for it is to open those two shells that are always so tightly closed for those animals. Um, and then in addition to bivalves and worms, hey, they eat dead stuff, right? So I like to think of them as the vultures of the sea. I think of them that way in a positive manner, i.e., they help to clean up, right? Dead animals, dead organisms, you know, everyone's in the food chain somewhere. So horseshoe crabs are extraordinarily important also for getting rid of dead animals as well as, and we'll talk about more of this later, but they're burrowing when they're either foraging or just if they're also hiding if a predator's in area. That actually helps to aerate the sediment, very important in the marine and estuarine world as well. So very important in our Indian River Lagoon, along with all of the other areas that horseshoe crabs are found along the Atlantic seaboard. In terms of horseshoe crabs, what we call the body plan, I always like to say, horseshoe crabs have to be unique. So for a lot of arthropods, right, a lot of those animals with jointed appendages like the crabs that we talk about, that includes insects as well. For those groups, usually you hear words that you probably heard before to describe their body shape and setup. So for crabs, let's take like an insect like a grasshopper. They have head, thorax, abdomen. Um, we've heard those words before, at least maybe. Of course, I've heard them a lot being a biologist, but yeah, sure, they're a little more familiar to us. Horseshoe crabs, on the other hand, when we go through their body plan, they don't make it easy on us. So you've been staring at this slide for a while. You probably see that these three sections for horseshoe crabs, we divide them in the prosoma, opistosoma, and the telson. So telson, hey, we had to have a unique name for that too, right? We can't call it a tail. So in the horseshoe crab, it's a telson. All as much as to me, their naming makes it more difficult. It's also more fascinating. Truly unique animals. So in terms of when we're looking at the ventral side, all of their anatomy underneath, horseshoe crabs have 5 pairs of jointed appendages, so jointed walking appendages, but then they also have those cute little feeder arms that I briefly talked about, which are located, if you're looking at that bottom picture, right in the center of the horseshoe crab towards the bottom of that graphic right there. You'll see these 2 little tiny feeder arms. And if you're looking at those arms, you'll notice that, well, where is the mouth on this animal? And the really weird thing about horseshoe crabs is that their mouth is located in the center of all of their appendages. So where all of those jointed appendages meet in the center and radiate out, that's actually where their mouth is. So are you all able to see my mouse? Hopefully.
Yes, we can see your mouse.
Awesome. Thank you, Jesse. That makes this this much easier. In terms of this is what I'm referencing. They have these 2 little tiny feeder arms. Those are called the chelicerae, right? Those are part of their unique anatomy. And right in between all of this is their mouth. Really odd placement for a mouth. If I'd never seen this animal before and I flipped it over, I definitely would not think that area was the mouth. really fascinating thing when we are looking at horseshoe crab anatomy closer, they actually have all of these little bristles that are essentially centered around that mouth. So if we look at a little better ventral photo here, this is what we're talking about, this area, right in between all of these walking appendages. Harder to see on this photo, but all of this is um, very jagged and abrasive. So horseshoe crabs, I always like to tell kids that they essentially chew with their elbows, right? A really weird way to eat. The other thing is they have to be moving, they have to be walking to actually chew their food. So they can eat, but they have to be exercising while they're doing it. So yet another fascinating thing in terms of for horseshoe crabs that's very different, so different with their body plan and their anatomy. Continuing down the line with weird things about horseshoe crabs, they have something called book gills. This is another thing that I get really excited about. And what are book gills? Well, they're circled right here in this photo. And if you all are lucky enough to see a horseshoe crab underwater, or if you Google some videos after this, you'll see that horseshoe crabs, when you look at them from the underside when they're underwater, the way they respirate is they actually have all these little flaps, that part that's circled there. And underwater, they look just like the pages of a book flapping around, right? So, okay, cool. Great way to name them. Why would they have that sort of respiratory structure? So, for horseshoe crabs, their book gills, the reason that they have all these layers is it creates more surface area. When we talk as biologists about respiration, we like surface area when it comes to breathing. So, if you think back to human anatomy in school, I remember they taught us that our lungs actually have all these tiny little pockets in them called alveoli. The benefit to that is within our lung, it creates a lot more surface area. We like surface area when we're breathing because we need it for gas exchange. The more surface area you have, the more oxygen you can pick up. Guess what? Horseshoe crabs use a similar strategy. Sorry about that. So horseshoe crabs use a similar strategy in terms of trying to maximize surface area, right? So I'm a horseshoe crab. I want to absorb as much oxygen as I can, especially important since I live in a water-based aquatic environment, right? Much lower proportion of oxygen in the water as opposed to us for air-breathing animals. And when we have all of these gills stacked up like that, like a pages of a book, it makes a really compact way for a large amount of surface area, a large amount of gas exchange, a lot of oxygen going into the body very efficiently for horseshoe crabs. In addition to all of that, horseshoe crabs can get pretty big. So I grew up here in Florida. I'm used to my horseshoe crabs being maybe the size of like a small dinner plate. But then when I talk to colleagues and when I give these presentations, I have folks from the Northeast, whoo, they always say, well, why are your horseshoe crabs so small down here? So interesting thing about horseshoe crabs, a trend that has been noticed is the further north you go, the larger they are, especially those Northeast populations. So when I talk about Northeast populations, one of the big ones for horseshoe crabs people talk about is Delaware Bay. Um, Immense and extraordinarily important population of horseshoe crabs up there. If I have time towards the end, I'll kind of go into the Delaware Bay story in terms of horseshoe crab conservation, which is very important. Um, but continuing in terms of what we're discussing, why are they so big the further north? I honestly don't know. I've heard different theories. I haven't read anything that's super specific about it. Doesn't mean that information isn't out there. It's a cool thing about science, right? We're always we usually get a lot more questions than we do answers when we do research, but it keeps us fascinated and it's extraordinarily important for advancement. Um, in terms of the last item on here, anatomically speaking, that we're going to discuss on this slide, is the telson or the tail, right? We gave it that fancy name. Telson, what's it used for? I have had many adults say, oh, that's where the stinger is. And I say, well, no, somewhat similar body plan to a stingray, but very different. I will admit and say that horseshoe crabs can look somewhat menacing or intimidating with their long, what looks like a spiky tail, as well as they have spines elsewhere on their body. They don't have any venom to hurt you with or anything like that. And as always with so many animals, I'm sure they're much more afraid of us than we are of them. We could cause a lot more harm than they could. Yeah, nothing on this animal that's there to even their little walking appendages underneath. They look like pinchers, but they're not even. Once again, they're not true crabs. Those little quote unquote fingers that they have, they're extraordinarily weak. They can't deliver a strong pinch that would hurt you, like maybe a stone crab or a blue crab, other animals that are true crustaceans, right? That have those true pinchers. So continuing into their anatomy, we're going to talk about some eyes. Like everything else, the horseshoe crab, very unique eye layout. So horseshoe crabs have a total of 10 eye structures. I use that word eye structure, that term, because their eyes are just so different from what we think about as eyes as humans. All of our eyes, they make pictures, right? Horseshoe crab eyes, they have 2 out of their 10 that do make pictures, somewhat similar to ours, but they have compound lenses. So kind of like, if you've ever seen a fly eye under a microscope, I always say it looks kind of like a stained glass window, right? And in the top left corner here, this is a really cool picture where this is a light that's being emitted through a molt. So this is like an exoskeleton. Snakes shed their skin, horseshoe crabs shed their skin as well. We'll get into that a little more later. But for this particular image, they're actually shining a light through the eye structure, the primary eye structure of a horseshoe crab here, the molt, and you can see all of those tiny little lenses. So to me, I get really excited about that. I think that's fascinating that they have that kind of eye, and I just think that that picture really like strikes me. And I've done that with horseshoe crab molts that I have around the office that I use for outreach. It is really amazing if you find a horseshoe crab molt. I keep saying molt, right? So it's not a dead horseshoe crab, it's just their exoskeleton. If you take one, you shine a light through it, and you look at that eye area, really just fascinating to look at, as you see. In addition to the 2 main eyes, this is where we get more into what I refer to as eye structures. So you have those 2 main compound lens eyes. And then in addition, they have some eyes that actually can sense and see ultraviolet light. So UV light spectrum, right? That's the really high energy light wavelengths that burn us, right? When we're outside and I'm working on a boat too long. We can't see that light, right? Ultraviolet, we can't see it. But guess what? Horseshoe crabs can. Really cool. They can see, what that means is they can see colors that we can't. Amazing. In addition to the UV eyes or the UV eye structures, they also have photoreceptors. So photoreceptor, if we break that word down, photo meaning light, receptor, pretty self-explanatory. Those eyes, they can tell the difference between light and dark, but they can't make pictures, right? So very different from their other kind of eyes, one that can see UV light, one that create actual images. For their photoreceptors, they actually have them in a lot of places on their body. Specifically, they have some on the telson. They also even have some underneath the body. So I always like to quiz students, and say, okay, well, if you're a horseshoe crab and you have all these eyes and they're placed all around, why would you focus, why would you have these eyes that are all they can do is just tell light and dark when you already have a bunch of other eyes? So The reason for that, or one of the thoughts, is that it aids them in terms of hiding from predators. So horseshoe crab, as we've said, they don't really have anything to fight back with, no venom or anything like that. So they are they're better at hide and go seek than us humans. So they burrow, they can hide really, really well. In addition to usually being very blending in very well with the substrate color, right? So they have that kind of sandy, muddy color. They also are extraordinary diggers. Makes sense considering that the front part of their body, that prosoma, is shaped like a shovel. So if I'm a horseshoe crab and there's a loggerhead sea turtle in the area, or maybe a shark, or even for areas where we have a mixture of saltwater and freshwater, like our Indian River Lagoon, right? Brackish water. You can also get alligators predating upon horseshoe crabs as well. So if any of those predators are in the area, one of the primary method for horseshoe crabs to avoid predation is by burrowing. So bringing it back to the eye structure or the photoreceptor topic, why would they have these all around their body? Sometimes when I pose this to students, someone gets it, and the the thought is, well, it probably assists them in knowing when they're completely buried, right? So they know if their eyes are, in multiple places around the body, if all of them see dark, right? If all of those photoreceptors just say, hey, everything's, dark, then they know that they're probably completely covered by the sediment and sufficiently burrowed to hopefully avoid predation. So, in terms of horseshoe crabs, you'll hear me say this, quite a bit. Such a cool animal, but we're only on the tip of the iceberg of what we know about them. There's so many questions, so many. In terms of marine animals, marine animals, we're always learning and or discovering new things. I always like that adage that we know more about the surface of the moon than we do the bottom of the ocean in many areas. And, even for those animals that we see, such as horseshoe crabs, there's still so many new things to learn about them. So continuing along this fascinating creature and its anatomy, let's talk about their blood. So folks who have, been interested in horseshoe crabs before, or maybe you heard this, in school, or, watched a documentary. Horseshoe crabs have blue blood. Okay. Their blood, as you can see on my slide, one of the most expensive things on earth. But first of all, we're going to talk about why is it so expensive. Before that, we'll talk about first why is it blue. So with the horseshoe crab blood, it's blue because they contain a protein called hemocyanin. So hemocyanin, let's break that down into something more palatable. What that mainly means is their blood actually has copper in it. So we have hemoglobin, right? That has iron. Our blood turns red in the presence of oxygen. That's the iron oxidizing. Okay, for horseshoe crabs, they don't have hemoglobin, they have hemocyanin. So, cool thing about them is instead of iron, they have copper. And when we start to think about, okay, blue blood, start to think about things that are copper that you've seen that have rusted, like a rusty penny. Um, their blood will come out if we extract it as a bluish to bluish green color. So we established why it's that color. But then in addition, they in terms of their blood, we actually use it biomedically. So my favorite fact to tell people or concepts that is extremely interesting about them is we use their blood in biomedicine. What do we use it for? Well, horseshoe crab blood has this interesting um, characteristic where, in the presence of really detrimental bacteria, so we call it in biology world gram-negative bacteria. That means bacteria like E. coli that can make us super sick. If horseshoe crab blood is exposed to that kind of bacteria, it actually has a visual reaction. It'll actually coagulate and change color. In terms of why is that helpful for us humans? Well, if you think about the biomedical industry, especially decades ago, before we had a lot of the microscope advancements that we have today, a really cool thing is when, we would have a vaccine or um, maybe like a dental implant or really anything that goes into the human body. One of the standard testing things prior to putting that in the human body, blood transfusion. We take a subsample of whatever we're putting into someone and we test it against horseshoe crab blood. We expose part of that blood transfusion, horseshoe crab blood, to make sure that it doesn't indicate that there's that bacteria present. So to make sure that it doesn't react and coagulate, showing that, hey, this is a tainted sample. So super important for us biomedically. Um, as a conservationist, I'm always hopeful of um, you know, advancements creating synthetic versions of horseshoe crab blood, or specifically the enzyme that we use as humans to do those tests. There are now a few different synthetic forms of it. But like any advancement right now, it's only produced on a small level and it's very expensive to produce. So horseshoe crab blood is still a primary standard in the biomedical industry. In addition to their blood, we already went over their eyes are amazing, but yes, as humans, we do, we study a lot about their eye structures and we're still learning about them, that can help to hopefully advance areas of, biomedical vision research and things like that. And then finally, my favorite fact that I feel like not many people know, and I only learned relatively recently, is the horseshoe crab shells. They're made of a structure called chitin. A lot of other invertebrates have chitin as well. But for the horseshoe crab chitin, they've actually done studies where they actually added chitin to a post-surgical stitches and or gauze or dressings because they've found that chitin, when used in those materials, when it's added, it can actually help to speed up recovery time. Why is that? uh I still have my theories about it, and I'm sure that plenty of people do. One thing that I did read, from someone who knows way more than I do about horseshoe crab chitin and substances, it actually has a really cool antibacterial property. So it kind of makes sense that, especially post-surgical, right, our primary thing that we're looking at is sanitization. We don't want our wound to get infected. So to me, it makes sense, that if it does have that property, that would be beneficial post-surgery. So horseshoe crabs, they do lay eggs. I hesitate to, but usually compare them to sea turtles when we're talking about the reproduction, because we have very familiar conception, especially in Florida, that hey, yeah, sea turtles lay their eggs in sand. Well, horseshoe crabs do that as well. That, right there is about that's where the similarities stop, and then we get all the contrasting. So horseshoe crabs, yes, they lay their eggs in sand, but they lay them in a different manner than sea turtles. So we actually, for horseshoe crabs, sorry about that, had a phone call coming in. So, in terms of horseshoe crabs, when they're reproducing, they do lay eggs in the sand, but horseshoe crabs, along with many other invertebrates, they have what we call external fertilization. So, what that means is the female actually deposits her eggs in the sand, and then the male actually fertilizes them after they're already in that wet sand. So, in terms of thinking about horseshoe crab reproduction, um, it's really interesting that external fertilization means that for one nest, you can have quite a few different uh, male and female inputs in terms of genetics. So, you know, there can be a female who lays her eggs multiple, males can fertilize it because it's external fertilization, and or if there are also adjacent females, they can lay their eggs in that same area as well. So you get all sorts of different genetic parental input in each nest, especially since horseshoe crabs primarily are what we call um, mass spawners or mass nesters. So they actually all usually spawn in concert. They actually have times of year, and I'm skipping a little bit just because I know that as advertised, I'm very long-winded when it comes to horseshoe crabs. But when we're looking at their reproduction, just to kind of speed up here, horseshoe crabs, they're mass spawners. They nest in these giant groups. And in terms of horseshoe crab nesting, fascinating because when you're lucky enough to see a spawning event, there's so many individuals on the beach. As scientists, these mass spawnings, besides being cool to see, is a great way for us to get an idea of horseshoe crab population sizes and things like that. So in terms of horseshoe crabs, um, and when we're looking at population sizes, and see on this slide, horseshoe crabs, they lay a ton of eggs. We think that sea turtles lay a lot of eggs, 100, 150 plus. Well, horseshoe crabs can lay anywhere from 2,000 to 4,000 eggs at a time. For those of us who are familiar, especially with invertebrates, a lot of them do this reproductive strategy where, hey, it's like, okay, I'm laying all of these eggs, right? And maybe a couple will make it to adulthood. Very similar to sea turtles as well. In addition, one of the things that I was mentioning was different nests, they can actually have multiple genetic inputs, meaning, one nest can have eggs and sperm from, many different horseshoe crabs. In terms of evolutionarily, this works really well for them. And I had said that they're mass spawners. So a big thing for organisms and animals that are mass spawners is they have to all reproduce at the same time. So I always wonder, like, okay, well, how do they know when they're going to reproduce, or how do we know as scientists when they're going to reproduce so we can, see these events and get more information about this animal? As with a lot of marine and aquatic organisms, their nesting is usually aligned with the tides. So in particular, horseshoe crabs like really, really high tides. So during the new moon and the full moon, that's when we have the highest high tides and the lowest low tides. Horseshoe crabs try to nest on that high tide. The reason is because they're laying their eggs in the sand, and usually a lot of the sand, if it's always underwater, our substrate, especially and including the Indian River Lagoon, a lot of times that sediment will be very acidic. Okay, so a lot of sulfur. Sometimes when it's low tide, we can smell the lagoon, of course. Horseshoe crab eggs, they're actually, that sulfur content, that's not the most hospitable or nice place to have developing eggs. They like the more oxygenated soil. So where would that be? It'd be in the really high tide areas of the sand. So those, horseshoe crabs, they prefer to lay their eggs while still in water. So it makes sense that high tide is when that gives them the access to that really beneficial substrate that has that high oxygen content. In terms of, I'll jump past why horseshoe crabs are cool and necessary because we already talked about that some. But yeah, once again, food web, definitely extraordinarily important in terms of quote unquote cleaning up seafloors as well as aeration. And then as a conservationist, I focus on things like that. Of course, outside of that, incredible biomedical contribution as well. And in terms of one of the primary benefits ecologically that I don't want to overlook here is horseshoe crabs and their relation to shorebirds. So I study horseshoe crabs. I'm also a huge birder. I love birds. For those of us who are bird nerds like myself, you may be familiar with a chunky, cute shorebird called a red knot. Very weird name for a bird, but red knots are actually, they're a protected species. So in particular, there's a subspecies of red knot called the roof of red knot. Their population numbers started declining later in the 1900s. And the really interesting thing is that red knots, they migrate thousands of miles to go nest up in the Arctic. So if I'm migrating thousands of miles and I'm flying along the Atlantic seaboard there, wherever I stop, I need a lot. And something that's been coinciding for millions of years is that horseshoe crabs, their nesting timeline, the primary time of year that they nest in Delaware Bay in that area of the US, coincides with when the red knot is migrating back in the spring. The red knot's heading north. So what's cool, and I like to connect for horseshoe crabs, is now several decades ago, some birders that would always go to Delaware Bay, they noticed, hey, we don't have as many red knots. And, that was disheartening to them. But they realized, well, we don't, we're seeing less of these horseshoe crabs too. Red knots eat their eggs. That's their primary benefit that they get from the horseshoe crabs. Horseshoe crab eggs, they're high energy, just like chicken eggs are for us. Extraordinarily important relationship where, the horseshoe crabs of Delaware Bay were a pivotal keystone component of red knot migration and success. So uh, eventually, these birders talk to the right people about, hey, our red knot's declining. I don't think it's helping that, we're seeing less and less horseshoe crabs, possibly because we are harvesting them for some of their blood biomedically, and other harvesting practices. So horseshoe crabs were actually then deemed protected and regulated in Delaware Bay. So long story long, horseshoe crabs actually gained protection and really just some of the first notoriety from birders, especially in Delaware Bay area. In terms of horseshoe crabs in Florida, bringing it local here for my last couple minutes. When we're looking at Florida horseshoe crab populations, and in particular Indian River Lagoon, we do horseshoe crab tagging. So there's two kinds of tagging that I'll talk about here on this presentation. We have visual tagging, and then we have acoustic tagging. So if we look at this picture of this horseshoe crab on the left here, it has what we call a visual tag. So tagging, as scientists and ecologists, there's a practice called, mark recapture or mark recite. If we want to know about horseshoe crab populations, if we tag a small fraction of them and then try to continue to get data over the years of when and where these tagged individuals showing up, it gives us a lot of information. So in 2015, FWC, Florida Fish and Wildlife, sister agency, state agency, DEP, they started Florida Horseshoe Crab Watch. Really cool. Up at that point, in 2015, we knew very little about horseshoe crabs in Florida. We kind of knew the least when you look at the entire Atlantic seaboard. And we also have much smaller populations in Florida. But interestingly enough, we started to notice, similar to the Delaware Bay birders, that, hey, we're seeing some horseshoe crabs. We don't know if we're seeing less. We don't know if we're seeing more. How can we start to look at population sizes and whether they're, growing, reducing, staying the same? And one of those ways is tagging. So in terms of the Florida Horseshoe Crab Watch tagging program throughout the state, there are multiple sites. It's an amazing program where there's a lot of citizen science, a lot of really great scientists working on this program and data. And in particular, one of the tagging sites that I want to highlight is Parish Park in Titusville, Florida. So if we kind of zoom into that visual tagging effort there, Holly Abeels with Sea Grant is amazing. She is in charge of that site for all of the visual tagging, and she gets a ton of data from the horseshoe crabs in that area that all goes into this Florida Horseshoe Crab Watch effort. So the reason I kind of focus on Titusville for the end of this presentation is, um, yeah, in concert with Holly and her studies and tagging in Titusville, we also, and in particular, this is brand new for Florida, we also actually have tagged 15 crabs with acoustic tags. So, I say acoustic tags like it's simple. I don't expect you all know what they are. In terms of acoustic tagging, I said visual tagging. So, hey, we tag this horseshoe crab. It has this little tag, has a specific number that you can read. On the tag as a website where you can actually say, hey, I saw, crab 12345 in Titusville on this date. And you can enter it onto the website on the tag. And, Holly and all of those scientists get that really valuable data of, hey, I saw your tagged horseshoe crab over here at this time, at this location. Gives us so many different parameters. But in addition to that, a really great way to supplement that tagging data is with acoustic tagging data. So why acoustic? What does that even mean? So acoustic tags, for visual tags, of course, the limitation is we have to have someone recite the tag. And usually that's when horseshoe crabs are on shore. Horseshoe crabs are usually only on shore when they're nesting. So definitely gives a lot of really valuable data. But then there's still a lot of data that we're still, is still out there about horseshoe crabs. In terms of when I talk about horseshoe crab populations and Florida Horseshoe Crab Watch and further studies, uh, for those of us who are sea turtle nerds like myself as well, uh, there was a time, and it's only recently we have more light on shed on this, that we called sea turtles. There's a time in their lives that we call the lost years. What that means is, if you think about us as humans, right? Us in Florida, we see horseshoe crab, or I'm sorry, we see sea turtles primarily at two events in their life. We see them when they are hatching out of the sand, right? And we're not touching them. We're watching them go down to the ocean, cute little sea turtle hatchlings. And then we're also seeing them when mama comes and lays her nest. Well, what happens in between hatchling and adult? For all of those in between years, they called it the sea turtle lost years. And thanks to some acoustic tagging and some satellite tagging, we actually now know we have a lot more light shed on sea turtles, quote unquote, lost years for those juvenile years, immature years, things like that. Why am I telling you all of this? Well, I try to get you interested by talking about sea turtles, and now we're jumping back to the horseshoe crabs. So, similar with horseshoe crabs, we see them when they're nesting. That's about it. Very valuable data in terms of when we see them, when they're nesting, when we tag them, when we recite tags. A great way to supplement all of that visual tagging data is with acoustic tags. So acoustic tags, they are these tiny little tags that actually emit a ping underwater. They emit a sound. I won't bore you with, the kilohertz and things like that, but all you need to know is it's a sound that's the frequency is not heard by us and most animals. It's outside of our auditory range, but these tiny little tags that we put on horseshoe crabs, they emit that sound, and what's really cool is that sound can be picked up by what we call acoustic receivers. So, thing about acoustic tags is you have something that transmits or pings, right? That's on the animal. We actually tagged 15 horseshoe crabs with these transmitters. So 15 tagged horseshoe crabs in this study that we've been working on. And then when we look at, okay, cool, we have the horseshoe crabs, we have 15 that are pinging. Well, we need something to hear the pings. So that's where receivers come in the Indian River Lagoon and focusing our study site, Parish Park, which, I introduced in Titusville, the one of the most productive nesting areas in the entire state of Florida for horseshoe crabs, we actually set up 3 different receivers. So you can see my Google Earth map here. This is a very entry level figure, but I don't want to bore you all to death too much yet. But you can see here that we have 3 receivers, and for these 15 horseshoe crabs that we tagged, were trying to figure out, those horseshoe crab lost years. So we know where they are when they're nesting. We know they're in that cove. Did they stay in that cove for the rest of the year? We don't know because we never see them. Do they migrate and move seasonally? Do they have areas that, they're always hanging out in outside of nesting? Do they nest on the exact same beaches every time? Do they have, nesting site fidelity, but also just site fidelity in terms of their ranges? So for the first part of this study, what we got from this data was, hey, presence absence data. When are they in this Titusville Cove? That's the study site. So at Parish Park. When are they around Parish Park? And then from there, we can extrapolate, well, how often are they using this cove? And in addition to the 3 receivers that we have, we also have data from other acoustic receivers in the lagoon from other projects. So this is the point where I have to use my best judgment and stop talking for a moment, so that I can hear some questions. But as you see and or hear, please stay tuned, because there is, we have additional findings. What we found is primarily, as you can see, and I put it on this slide in case I got long-winded, so you're able to see it, etc. We have found horseshoe crabs, they go all over. Like, we didn't know if we would have, oh yeah, 10 horseshoe crabs stayed in the vicinity of these 3 little receivers, for most of the year, and maybe 5 roamed. All of the horseshoe crabs, roamed throughout a large area of the lagoon. And while I take questions, I'll show you that broader scope of map. So you'll see there, I've now zoomed out of Titusville. You can see just initially here the northern and southern boundary for the tagged horseshoe crab ranges. And just for your reference, that's about 18 miles. But they actually, we're sure that they go beyond that as well. The limitation with acoustic tagging is if you don't have a receiver, you won't have data, so you won't know where they are. Plenty of next steps in this project. We do plan to re-tag later, and see, even more data, right? Like we're just starting, just the tip of the iceberg, like I said when I started this presentation. But fascinating animals. I appreciate you all staying with me, and I will answer questions if we have a minute, or please email me. I'll put that up right there. Thank you all.
Fantastic. Thank you so much, Kirk. This was a great presentation. I learned a whole bunch of new things about horseshoe crabs, and I thought I was pretty well versed in them. So that was fantastic. We did have a couple of questions come in. So why do you think we don't find horseshoe crabs with the same occurrence throughout the entire lagoon?
That is one of the $1,000,000 questions. And in
That's
particular,
why we're tagging them, right?
yeah, right? Yeah. But in particular, the merit of that question. So I ain't go over my range, but our office covers the entire Indian River Lagoon, right? So just like the NEP, they're superheroes. We try to be superheroes too. But my office is in Fort Pierce, so more of the central and southern part of the lagoon. Well, I have to go, I have to drive almost 2 hours to my study site in Titusville, right? But exactly what the question referenced very accurately, I don't see them in Fort Pierce very frequently. I will find some molts, so there's evidence that they're here, right? Because I found a mold, I found an exoskeleton. But beyond that, we're still trying to figure out, exactly kind of what Jesse said, like, why are they not here? And or I always like to say, where are the horseshoe crabs? Where are they? But also, when are they? So for that small study area with, that our initial study, that we still have plenty more, extrapolations to make from the data. Like, yeah, where are they and when are they there? And are they traveling all the time? Are they always in the same place? We have found, though, initially very mobile. Sorry, that was long-winded. Well, Any other questions?
Yeah, no, I thought that
I'd
was
be happy
great.
I can answer. Yeah.
I mean, that is one of the big questions is why do we see them some places and not others? Why do we see them during some parts of the year and then when they're supposed to be here, they're not? And our IRL crabs are very weird. That is the one thing I tell people. They do not follow what everyone else does, which I like them for that, honestly. So why do you think it is so common to see so many dead horseshoe crabs on the beach, especially during nesting season?
That's a great question. So, believe it or not, as we said, let's paint that frame again of mass spawners, right? So, horseshoe crabs, on average, and I'm sure it you know, it differs per location, but from what I've learned from folks who know way more than I do about horseshoe crabs, they can have anywhere from like 10 to 20% mortality during nesting events.
Yeah,
So, if we think about mass spawner, especially in the Titusville, at Parish Park, with Holly and her crew, like if you're getting hundreds and hundreds of crabs spawning, 10 and 20% is a big number to us, right?
right.
To find. So many times when I started looking through years ago, um, I was looking through some like the FWC fish kill data from over the years, and there are many reports of I just found 10 dead horseshoe crabs, right? Um, and for me, I get excited about that as a scientist because I say, oh, well, at least we know they have some horseshoe crabs there. I'll take it.
No, I do too. I'm like, I always say it's not a bad thing that we're seeing dead horseshoe crabs. It means that they're here, and that is so good to know, you know? Yeah.
Very true.
So in regards to your acoustic tagging project, why were only 15 horseshoe crabs tagged? Was it funding or where it's there a statistic that was significant? So why 15?
So 15 money. It's an
Okay.
easy answer. It's funding. For Yes, and much more expensive than the tags are the receivers. So those receivers are very expensive. I when I deployed those receivers, I had anyone who knows me, and Jesse knows this, like I always have a contingency plan. I'm always thinking like, what are three ways that this might go wrong? And I've had colleagues who lost receivers. They
Right.
lost thousands of dollars of equipment. Like just, I mean, you're putting it in the lagoon, you're putting it in the water. You gotta, it's definitely precarious. So in terms of those receivers, I actually had like three different anchoring systems. So I had a redundancy system where like, well, if the chain fails, at least it's still on the PVC that's in concrete in this, in the
Exactly.
lagoon floor.
Yeah.
Yeah. But definitely funding. I know that that's not like a fun answer, but
Accurate,
it's
you know,
definitely
and I think that's something that a lot of scientists and researchers struggle with, is finding funding for these different projects. And it's nice to know that, that can be one of the reasons why we can't go farther with this research. So if you want more funding, I hope you get it. Can we volunteer to monitor horseshoe crab populations?
yes, depending on your area, there are multiple Florida Horseshoe Crab Watch coordinators. So in terms of in Titusville, like I said, Holly Beals, and she knows the most about visual tagging sites in Brevard. I'm down here in St. Lucie County, so it's definitely always a county by county basis as to like who we would refer you to. But for those who are specifically interested, please email me and I will happily connect you to the right place. If you don't mind, just email me where you are
And if
too,
you're
because that helps me.
Yeah, and if you're up in my region, up in Mosquito Lagoon, Marine Discovery Center is the one that does the horseshoe crab nesting near us. So you can contact Abby. She would love to have you come out and volunteer with her. Yeah.
Yeah, and I would be remiss if I didn't say Jesse was actually the person who helped to get me into horseshoe crab research in the IRL. Always appreciate that.
I love them. So that is actually the perfect segue into our last question for the day. You do not
Sure.
have to answer this because the answer is it's not going to happen. But I
Oh,
got you into horseshoe crabs. So we have someone who wants to know, how do we get Jesse to like birds as much as you do? And if anybody knows me, you know that I do not like birds.
and Jesse knows me, and she knows that I just don't understand that about
I
Jesse.
Sorry,
my
man,
gosh.
it's not gonna happen. The only
mean-
birds I like are the Philly Eagles. We all know that.
Oh, well. Hey, come along with us and tag some oyster catchers, and you might feel a little different, but
Maybe.
yeah, I mean, we can always hope, you know.
Well, Kirk, thank you so much for coming by today. We really appreciate it. If you guys have any follow-up questions, please feel free to reach out to myself or Kirk. We can get you in touch with the right folks for volunteering. Just a reminder again that this webinar will be available on YouTube, and we do appreciate it when you share it. So please go ahead and share that. And we will have another webinar coming up in early May. So keep an eye out on your email, Facebook page, and all social media, as we will be posting that information as soon as we have it available. Thank you guys very much. Have a great rest of your day.
Thank you, Jesse. See ya.
See you, Kirk.